Okay, cool. So we're live now. It's okay. We'll remove this initial part, so we can just chit chat. I wonder where Mafe is at this point. So we already have 11 viewers. So that's good.
Hey, viewers.
Hey, viewers. Where are you looking? Okay, so let's switch to Spanish just to start. Buenos días para todos. Muchas gracias por acompañarnos en este evento de Python Bucaramanga, que vamos a estar haciéndolo en inglés para tener a nuestro primer invitado internacional. Cuéntenos un poco desde dónde nos acompañan el día de hoy. Y pues ya vamos a empezar dentro de poco. Yeah, I was just welcoming them and telling them to share where they are located and some random stuff. Cool. Cool. We have greetings from Bogota. How are you doing, Ernesto? Long time no see. Hey there, Jorge. How you doing? Oh, we have people from Iran. That's cool.
Hello.
Hi, Amir. Hey, Diego. How you doing? Don't be shy. Tell us where you are. No sean tímidos. Díganos dónde se encuentran. Okay, we'll wait a couple of more minutes and we'll roll with it. Okay. We have Mafe here. Hi, Mafe. How you doing?
Hello. Good. Hello, Tyler. Nice to meet you. Thank you so much.
Yeah.
How's it going? Everything is all right?
It's good, yeah.
Okay, good.
Hey, Augustin. Good to see that. I have a familiar name here.
Cool. We have some people from still Bucaramanga, people from France, Chile, Buenos Aires. Hey, Agustin, he's saying hi to you.
Yeah, we go way back.
You go way back. Cool. Nice to see friendships last long time.
Ivan Pulido.
Yeah, Ivan Chipchombian from New York. How are you doing, man? How's the apple going?
Dryer.
Cool. We are reaching 20 viewers. Let's wait a little more. And if you know people that might be interested in learning more about Python microscopy and napari, please send them the link. So more people can enjoy this awesome talk that we're about to have. Cool, man. Good to have you here also.
How is everything? Where are you now? In what state? In DC?
Me?
Yeah, you.
Yeah, I'm in Boston, Massachusetts.
And how is the weather? Really hot?
It's beautiful out, actually. It's a lovely day.
Yeah.
It's a lovely day to be indoors.
I just took a little walk, get a little air. It was,
Will you take one, or you already took one?
Already took one, yeah.
Okay, that's cool.
How was it
Cool.
down there?
Okay, so almost there. Let's wait a couple of minutes more, and we'll start in a second.
Mafe, what city are you in?
Bucaramanga.
You are, okay.
Yeah, it's really hot.
Is it?
I know right now in America it's really nice weather because, June or July is like hell. It's super hot. It's impossible to enjoy the weather, but now it's really, really cool. September, October.
September's good.
Yeah, it's fresh and you can have fun. You can go on bike ride and you can do hiking.
Yeah.
It's really cool.
Awesome. Cool. So I think we can start now. It's 5 for the hour. We have 20 people around here, so I think that's good. Let's begin. So welcome everyone to our 6th event of the year. This is a special event. We're trying to change things up a little bit and have more guests from everywhere now that we're doing all these things virtually. So the plan is to keep doing this regularly every month. So the next month, hopefully until the end of the year, we'll have 2 events per month. One will be with local guests, other one will be with international guests. They may or not be in English, depending on if our guest speaks English. So this one is in English. And so, yeah, I mean, I know that there is a lot of difficulties here in Colombia. Not everyone speaks it or understands it, but this is also like trying to go into the direction of it's important to learn it if we want to work in technology and anything else, especially in academia. So we'll try to share some resources in the coming weeks, so where we can improve English, where you can study for free, and some also like tips on how you could go around it if you still feel like you can understand but you cannot speak it. Well, we'll try to give some tips on how we can improve that. So yeah, so I should have presented myself first, but Gonzalo. I'm located in Bogota. I'm co-organizer of Python Bucaramanga. So yeah, please introduce yourself and then we'll go from there.
Okay, hi guys. My name is Mafe Toro. I am from Bucaramanga and I am part of Python Bucaramanga. And we are very excited and really grateful with everybody to be here. I know it's really a challenge for me too because my English is not pretty well, but I think it's really good. We try these new skills for grow out, and I think it's really good. Start, try to be thinking like really IT. In this area, the IT, we need the English. We need to improve our English. We need to improve our skills. So I think it's really good we start with this idea, and I hope everybody can really enjoy this really amazing idea. We had so really good news with Python Bucaramanga. So really, I say thank you everybody for being here, and thank you Taylor for being here.
Yeah, Tali, just let us know where you are.
Sure, yeah. Hi, my name is Tally. I am in Boston, Massachusetts, in the U.S. I'm thrilled to be here. I really appreciate the opportunity to come talk to you.
Cool. So maybe some context here, like part of my day job with Quantside is working on a project that is Napari, that is part of something that Tally is going to be talking today. Tally is a core developer in that project for, I don't know, how many years now?
2 years.
Like a couple of years. So I've been working with Tally for the past, I don't know, since February, I believe. So, sorry, Mafe, can you put mute? Is it you? Thanks. Yeah, kids and dogs, they make noise. It happens. So yeah, so we'll be chatting about tools in the context of like microscopy. Our very special guest, I know Tally for a couple of months now. I'm very happy to have met him. He's an awesome scientist, but he's also an awesome coder. And he has a PhD in neurobiology from the University of Washington in Seattle. And is currently a lecturer and microscopist at the Harvard Medical School. And I'm super thrilled to have him here. Let us know a little bit more about what some basics of microscopy and how are the challenges that we are currently facing in this area and how Python can come to the rescue. And of course, Napari, which is an awesome tool that he's helping develop. And I'm also super happy to help also improve. So Tally, the floor is yours. Take it away. Just a reminder, sorry, for everyone who has questions about anything. If you think you would like to hear more about a specific topic that Tally is discussing, please write on the comments and we'll jump in the conversation and try to tell Tally to maybe expand on something if you want to know more. And as usual, keep the questions going, and at the end, we will try to answer as much as we can. So
Awesome. Thank you, Gonzalo, and thank you very much for your work on napari. We've benefited greatly from your work the last few months. So, and thanks for the invite here. Yeah, I will. Let me share my.
Yeah, go for it.
Let me just re-emphasize and request that if you have any questions, it's too bad we can't be in a slightly more casual face-to-face kind of environment. But I will encourage Gonzalo to stop me based on whatever comments you put there. So feel free to do that. I am going to tell you a little bit about what I do. I'm not going to dive too much into the microscopy, just enough to give you some context for some of the specific data handling challenges that we have. And after kind of presenting the challenges, I'll just talk to you about napari a little bit, just to introduce it. It's a multi-dimensional image and data viewer written in Python and for the Python environment. And I'll also talk about really sort of one specific usage that I think I use a lot that I think is pretty key for my stuff, which is pairing napari and Dask. Dask is another Python library to sort of wrangle out of core data that is too large to fit into RAM. So yeah, I'm Tally Lambert. I am a microscopist and I work in a core facility at Harvard Med. If you haven't heard of a core facility, this is basically a shared facility, a shared resource where I basically help all the researchers, postdocs, and grad students to design and execute their microscopy experiments. So we have a lot of microscopes. We tend not to do the experiments for them. We train them to do them, but our general point is to be local microscopy experts. Personally, in the core, I have a couple advanced instruments. These are sort of quasi-commercial, newer microscopes that have sort of newer capabilities, but perhaps less easy to use, and I help people use those. But what I really like to do is code, particularly in Python, and I like to facilitate common data pipelines that we have. So a lot of the people who are using our microscopes are just core biologists, and they have kind of other things to worry about than figuring out how to wrangle all their data. So it's fun to sort of take what we know about handling data and try to grease the wheels or make a pipeline that is a little easier for them to use. So that's kind of how I got involved in all of this. I'm going to just give you some context on microscopes just to make sure we're all on the same page. Not too much great depth, but just because it's if you don't know this, the rest of it kind of loses some meaning. So, again, microscopes, they form an image, magnified image, and we basically, we use lenses and project it onto a digital camera. I'm sure you're all familiar with digital cameras. But the point with a digital image is that essentially, we convert some optical image into an array of numbers. So if the image coming out of the microscope is projected onto a camera, each pixel on that camera is going to count the photons and convert it to some number. So that's our core data, basically. It's just a single image is a two-dimensional array of numbers. But those dimensions, that single image can quickly get higher dimensional if we take, for instance, a focus stack. So take an image, change the focus, take an image, change the focus, take an image to try and capture three-dimensional information about an image or a sample rather. And I'll get more into higher dimensions in a bit. But the next step really is, so we've got these fancy microscopes. They pump out, gigabytes of arrays of numbers, but then we somehow need to extract meaning from that, biological meaning that the biologist can use to report something that they're studying. So for instance, you might have some noisy image of, in this case, these are just like nuclei in cells, and somebody might want to count those cells or say something about the area of the nucleus or the volume, make some measurements, etc. And you can do all that manually, but it's much, it's less biased to be able to do that in an automated way. And of course, we can do a lot more data throughput if we can somehow automate the process. So this pipeline really is at the core of everything we're talking about today. We capture data in digital, multi-dimensional arrays, and then we need to somehow extract that information. So that's the core challenge. And the classic toolkit here for quite a while, which is just still the bread and butter of what we use to do this, as long as we're in Python at least, is NumPy, scikit-image, and matplotlib. So NumPy, I suspect a lot of you are familiar with. NumPy is the core, well, it's a third-party library, but it's sort of the main array library in Python. If you're working with just arrays of numbers in multiple dimensions, NumPy is the main library for that. Matplotlib is an incredible plotting library. It sort of started, I believe, out of the desire to make something somewhat MATLAB-like in terms of its graphing interface, but has evolved to be just a really awesome general purpose graphing and plotting library for Python. And then there's scikit-image, which for me at least, I use a lot. It has just a library of functions for doing image analysis. So I'm just showing some basic code here that might be typical here. So we would import matplotlib and TIFF file. TIFF file is just a module that lets me read a TIFF image. And so we would just grab that data. That data is now a NumPy array, and we can just hand that to matplotlib. And so there's like an image somebody might have taken on a microscope. And then the next step generally is some sort of processing. So for instance, from scikit-image, we might import some filters. And here I'm just making a mask where, show me all the pixels in this image where that pixel is greater than some number, and that threshold function gets that number. So details here don't really matter, but the point is, this is sort of classic stuff. But the real challenge here comes as the number of dimensions start to increase. So what I'm showing you here is just a 2D image, and it's easy enough to kind of count stuff and see stuff and interact with your data there. But as the dimensions start to get higher, it gets much harder. So, just to clarify what I mean by dimensions here, X and Y obviously is our 2D spatial dimensions. As I said earlier, you might take a Z stack of focus, changing focus and taking an image so you can capture 3-dimensional information in space about your sample. But then you might do that every minute to capture a time lapse. So now you might have a 4-dimensional data set of some 3D object over time. And then channels are a very common thing. So we can label our samples with different colors, for instance, to show something, perhaps a different protein in each color if we wanted to say how are these different proteins interacting in the cell over time. So now we got 5 dimensions, and you might do that at different physical locations on a cover slip. Now you got 6 dimensions, and there are more sort of domain-specific things that might increase the dimensionality of your data. And now this sort of old paradigm of having, plot im show, single image, it works, but you need to start building in additional, you have to project higher dimensional data down to a lower dimensional space so that you can kind of even work with it. And that just kind of gets limiting. So just to give you an example of a higher D data set, this is just an image taken on one of these sort of fancy microscopes I'll tell you about in a second, of a cell undergoing mitosis. So this is a 5D data set of a 3D cell in 2 colors over time. And in this particular one, we're looking at mitosis. This is cell division. So these are the chromosomes that pulled apart there. And somebody might want to, measure the shape of these chromosomes, say something about their dynamics over time. We want to be able to do that automatically. Some of the modern scopes we work with can really generate quite a bit of data. So the ones I'm specifically working with a lot are called light sheet microscopes. I'll give a brief aside on that just because I think it's kind of interesting, and but I won't belabor it. Anyway, we can quickly generate tens or even thousands of gigabytes in a day, or hundreds of gigabytes certainly in an hour or so. That's the raw data, and then very often that needs to be pre-processed in some way before you can even view it. There's certain transformations we need to do to even be able to look at it. And then the processing begins, and then there's more, data explosion, if you will, for that to even to happen. Later in the talk, I'll give you a quick demo on how we can use Dask. Dask is a array library that I'll talk about in a bit. But Dask and Napari to make some of this handling of larger dimensional, bigger data easier. Just because I mentioned light sheet, I'll tell you what it is. Just because it's a pretty awesome development in microscopy over the last 20 years, and so I'll just tell you about it. So wide field microscopy, this is the jargon for what we just mean for standard microscopy. This is, if this is a cover slip here, this piece of glass, and this is just sort of a cartoon of a cell, we usually illuminate from the bottom, so the lens might be here, and we illuminate the entire cell with light. And it's got some fluorophores. These are little organic molecules that absorb that light and give off light again. Now we've got some lens and it's forming an image of this, but the thing is we can't differentiate between the light that came from in the focal plane in the cell and outside of the focal plane. So just like with a regular camera, obviously the background gets blurry, right? And so that's what we have in microscopy is we have the good in-focus stuff superimposed on the out-of-focus blurry stuff. And if you sort of think of that as signal in focus and background out of focus, you basically have a poor signal to background ratio and your images look blurry and they're hard to analyze. We have ways to deal with that. There's an old workhorse type of microscopy called confocal microscope. And a confocal microscope is clever because it can basically reject light that came from out of the focal plane. So if we go back to this example, let's say we illuminate the whole sample, but now with some clever optics, we can put a pinhole somewhere in the light path and basically throw away the light that came from outside of the focal plane. So now you get a nice sharp image because you have no out of focus light, but it's wasteful because you've still illuminated your whole cell. And it turns out that cells are sensitive to light. So you're kind of killing them all the while, and you're kind of wasting the fluorophores, these things that are absorbing light and emitting. They don't last forever, and you're kind of wasting them. So the point here is that light sheet is a new technology that's clever, brings in a second lens, and does this, sort of illuminates only the plane you're interested in. And so that's the best of both worlds, because we can collect all the signal from our focal plane without wasting anything from the background. So that's the key idea with light sheet. But these microscopes are they don't look like what you're used to thinking about for a conventional microscope. This is an example of one. But they do generate some pretty amazing data. So the one I showed you before was collected on this type of microscope. Here's another one from the Betzig lab showing an immune cell, a T cell and its target. This is what they call the immunological synapse, where your immune system is at work. And we can now study these things just in incredible, detail, multidimensional detail over time. Here's another example of viral particles. So this magenta thing here is the nucleus in the cell where all the DNA is. And every green spot here is a little viral particle that has been generated. And this person was studying how do they move around in the cell and get it secreted. So again, we can start to ask questions in incredible detail and perhaps go in there and analyze this. Here's an example where we might want to track these things over time. So this is this was not done in Napari, but this is using a program called the Mars. But this is a pretty typical thing we might want to do, which is just to say, find the spot, watch it move over time, and then let's say something about its velocity, etc. Right? We just want to extract those numbers. And some more pretty images. I will assume there's nothing in the chat, Gonzalo, because I can't see it at the moment. But
there is a question right now, but maybe we can answer it right now. Why not? So Ivan is asking if you mentioned it and he missed it, but what resolution can be achieved with light sheet microscopy? Yeah,
good question. So there's nothing specific about resolution in light sheet. Which is to say, the resolution one can achieve in light sheet follows the same rules as conventional microscopy, which is to say it depends on the color of light you're using to image, and it depends on the lens you're using to form the image. But if we want numbers, we're still sort of limited in the best case scenario in mid 200 nanometer range, which is to say, you know, as soon as things start to get closer than 250 nanometers, we can't really tell them apart. So we're still roughly in the same ballpark of resolution. The one thing that does change is axial resolution, or Z resolution. So if I'm looking at sort of two objects down the optical axis of the lens, I can begin to tell those two apart much better in light sheet. But roughly speaking, 250 nanometers is still an upper, you know, a best case. Yeah, so this is just another example of data analysis. So that's the appealing, right? You see these amazing images and they're beautiful and people come in and everybody gets very inspired. But then at the end of the day, if we can't leverage that to make some sort of meaningful biological finding, it's mostly just inspiring for donors to give us money, but you know, we're not really doing anything with it except looking at it and saying, ooh, which is good, but we need more. So my work in trying to help local people deal with all this data started with just now I think a rather crappy data processing tool, but it was just sort of meant to be kind of a user interface for people to drag and drop their folders on there and do some of this pre-processing that is required to handle the data coming off of there. And this was helpful, but it had some downsides. It's slow. Basically, you have to wait for the entire experiment to be processed before you can even kind of look at the first time point. There are ways sort of around that, but that was the standard way. It was greedy in the sense that, you know, it did everything. It would do all of the processing one might possibly want to have done, and it wasn't smart with the way it did that, and it was definitely not very interactive. You couldn't do something and change it as you were watching. And so that's why I got interested in Napari. Napari started before me. Juan Nunez Iglesias and Loic Royer they conceived of it and started it. A number of people have worked on it at this point. Well over 70 people have contributed, and I've been working on it for a couple of years. So Napari is an n-dimensional viewer, a multi-dimensional data viewer. It's open source, community developed program. And yeah, it's really designed to, you know, visualize multiple dimensional data sets that may be too big to handle in RAM. And it's a work in progress. We still, you know, we certainly have a lot of, on the analysis side of things, work to do, but we're trying to just sort of create a hub in the Python environment where, you know, Python already has an incredible wealth of analysis libraries out there. And it just sort of, one could want a better way to sort of integrate and view all the data as you're working with it. And that's what we hope to be. So yeah, we want to be a fast interactive visualization tool for n-dimensional images and data sets. It's really important for us to integrate into the existing scientific Python ecosystem. So that for us means things like NumPy and Dask, which I'll talk about. I gave Gonzalo some links that he can post, but I'll get into more detail there. And SciPy and scikit-image, and perhaps a lot of these machine learning libraries, because a lot of the most interesting stuff in image analysis is coming out of machine learning these days, and a lot of that is done in Python. So we really just kind of want to stay in that ecosystem. And of course, all the core developers also just like Python. Right, so that's the motivation. I will say we lean very heavily on the shoulders of some giants. So VisPy is a library that we use to do all of the OpenGL based rendering. So all of the fancy movies you see of things rotating in 3D, we can thank VisPy for that. That's an incredible library. NumPy obviously and Dask are the key array libraries, and the interface is built using Qt, PyQ. So this is the viewer. It kind of has a Photoshop-like layout in the sense that there's a canvas where, you know, all of your data is shown, and then we have this layer list. I'll talk about layers. Layers are a key concept in Napari, where we have different sort of canonical layers to handle specific types of data. So this is an image, so we have an image layer. But you might have points for coordinates in space, or surfaces, etc. And I'll talk more about that. Each layer type has some controls to control commonly used things for the way they're displayed. And there's dimension sliders and some other stuff. I won't go into that. But yeah, layers are really the key concepts in napari. It's possible that some of you work with data that might fit into one of these layers, even if you don't work with images. So I'll mention all of them just in case. So like I said, yeah, the image layer is the one that I use the most. This handles 2D, 3D, but multi-dimensional data, and with all the standard kind of display tools you might want, like contrast and color changing and rotation of your three-dimensional view and stuff like that. One increasingly common way to store really, really big data is in what's called a pyramidal format, or a pyramid image. This essentially, like, let's say you have a, millions of pixels by whatever, some massive, massive data set, a really giant image. It's very hard to view all that because that's just, it's huge, right? So you downsample it into smaller and smaller versions, and then you ask the computer to basically pick the lowest resolution version that you can use to view it at the zoom level you're asking for. So this is exactly how Google Maps works, right? You zoom in and you get higher and higher resolution. So we can support that in napari too. So it will load just what is necessary to show you. So a quick aside here before I talk about the next thing is I do want to talk now about the Dask versus NumPy, because Dask is amazing, and if you're not familiar with Dask, I think it's worth being aware of, particularly if you work with arrays or with pandas, data frames. It has a really nice API around those things. So NumPy, the classic array library, might do something like this. You import NumPy, and you could create an array or read in some data as a NumPy array. And the main point here is that this is in-memory array. So when I create this array, it gets allocated into memory immediately, and I can access it here like that. So it's and if I want to create a 100 gigabyte array, I better have a big computer, or I need to come up with some other strategies. One of those strategies is Dask. So Dask basically wraps NumPy's API, so it has the exact same API, well, not the exact same, but it reproduces much of the NumPy API surface, but it does so representing the array as a sort of chunked underlying array that you can access individual chunks of that larger array at any given time. So for instance, and so it's all kind of lazy on demand, if you will. So here, if I repeat this example from NumPy, but I say do with Dask, if I import Dask array as Dask array, now if I create a random, a 4x4 random array, if I look at that, you'll see it didn't show me the numbers. It shows me that this is a Dask array. It's sort of a representation of an array. I can see the shape and the data type and how big the chunks are, but I haven't actually taken up any memory yet, and I haven't actually generated any data yet. And that actually, if you could see, would have happened immediately. And until I call compute, it doesn't actually do it. So when I call compute, then the random generation happens in this particular example, and then the memory is consumed. So you can do this also with indexing, for instance. So if I have this array here, a 4x4 array, and I ask for the first row, there too, I've gotten a new Dask array that hasn't actually computed anything until I call compute. So this is the for us at least in napari, this is the main feature of Dask. And what it lets us do is it lets us queue up the concept of loading, processing, etc., without actually doing anything. And then napari can basically, as you move around the sliders, I'll show you here, napari can ask Dask to basically execute what is necessary to give you that chunk of data. So this is that data set you saw earlier with the cell undergoing mitosis. And I haven't really explained yet the processing that is necessary. I will in a bit, but the point is you can see as the slider moves here, there's it's basically doing a transformation, and it's also doing some deconvolution to make the image look crisper, but it's only doing it once I ask for that specific time point. So by contrast, the original way I would have done this is to collect the data on the microscope. Let's say I got 50 gigabytes. I would then do this de-skewing operation. I'll explain that later, but just pre-process the image so that I can view it. That takes up another 100 or 200 gigabytes, and I've got 250 total. And then I might deconvolve it to make it crisper, more contrast, so that I can do some segmentation. That's another 200, right? So I've taken my 10 or 50 gigabyte data set, and I've turned it into a 500 gigabyte data set just so that I don't have to do it again. I've done that quite greedily. So here, basically, I'm just going straight from the raw data set, and only when I get to that time point in the movie does it load that file, do the processing, and show me the result. There's obviously a little bit of a hit in terms of performance. It doesn't play the movie as smoothly as you would if you had done it up front. But that's fine in most cases. And then when you find that data set that's just perfect, and you need to perhaps render the movie that you're going to put in your presentation, then then obviously you do it. But this lets me save a lot of space ahead of time. So at the end, I have a notebook where I'll just go through that example, and I'll share it with you if you'd like to. Let's see, how am I doing on time? Pretty good. So the other layers, I'll kind of breeze through these. Labels are just a way to, it's an array of integers, basically, to identify a certain pixel in the image as belonging to some class or identity, basically. So, here we might segment these coins in this image, and all we're doing, we have tools to paint labels onto the image, but you might do this in an automated way, of course, as well. Or you might even do it in 3D. So this is a brain that's been segmented to show different parts of the brain as being a certain part of a certain region of the brain. Points are obviously huge. Most of the time, we're going to be identifying some coordinate in space that represents something interesting happening. You might manually annotate these, or you might use some analysis to do that. This is one of the places where VisPy, that library I was talking about, really shines, because this is, I think, 1 million or 2 million points, each of which has a different color and size. And I'm just rendering this on, in this case, this was just my MacBook Pro, and it does a great job. So that is a pretty great part of that library. So that's points. Vectors, obviously just a vector in space representing some orientation. Tracks are a very common thing. So like I was saying, in microscopy, we might want to follow some object over time. So that's basically, in every time point, we have to identify what is an object. Okay, so this is one, this is one, this is one, this one. And then in the next time point, somehow associate each object with the next, where it is in the next frame. And a track is a way of representing that provenance, that object over time. And so these little trails you're seeing in this image here show where that object came from over time. And that's another particularly useful layer for biologists. So here, for instance, this is a C. elegans, a little nematode, a little teeny worm under developing, and its nuclei are dividing, and so people might trace the lineage of dividing cells over time. And this is a pretty cool plugin made by a guy named Alan Lowe. Surfaces, these like the surface of a brain, or any surface you can represent with a series of vertices. We can show those, and they also have. This is an example of like a 4-dimensional surface. So a surface which over time has some intensity associated with it. So in this case, you might have a MRI or a functional MRI showing blood oxygenation or activity over time. So that can be superimposed here. And shapes. So shapes are just, yeah, you know what shapes are. Vectorial stuff like you might get in Adobe Illustrator or something. And one of the nice things here, so in that first pipeline I was showing you with scikit-image and matplotlib, for me at least, one of the key tools was always IPython and perhaps Jupyter Notebooks, where you've got your image there and you've got your notebook open where you can interactively work with the data So that's obviously critical, and so that's a very important thing for us to support. So here's an example, I'll go back to the beginning, of some interactive data analysis. So the movie starts just with that image I showed you. So here's just some nuclei, and this is actually the example I used before. So I'm going to threshold, the layer is my my image layer, and I'll get some threshold, so some number that I'm going to use to separate the nuclei from the background. And then I'm going to add to the viewer a new type of layer, a labels layer. This is that integer type layer. So here I had my original image, and then I added labels there. Oops, sorry. And the main point I wanted to make here was that right here, I will then add points manually in the graphical user interface. And we can access this sort of in two ways, right? So I can get it from the code. I'm getting the points data there showing I have an empty points layer, but now I go in and I manually mark some spots that are of interest to me. And now I can go back to the code, of course, and see what is my points information and how many points. And so that's the built-in console that lets you do interactive analysis. And of course, like I said, it's important for us to integrate with scientific Python ecosystem. So this is an example of running some machine learning with PyTorch to do number recognition, watching the prediction update over time in napari as we watch the loss function go down as the model, the machine learning model trains itself. Okay, so yeah, I definitely want to point to the people who are doing a lot of this work. So Juan obviously was one of the originators. Juan and Loic started this out. Kira did a lot of work early on to establish some of the proof of principle way of representing the data in BizPy and stuff. Nick jumped on early on and has done more commits to the repo than anybody, and he is essentially our fearless leader in meetings and stuff. He's incredible, and I've been on it for a couple years. But like I said, so many people at this point have contributed. So thanks, obviously, to all of them. So I'm going to move into demo mode now, but I'll also pause just to see if there are any questions at this point.
Yeah, there are some questions, so let's just go ahead and answer them. So let me see. Yeah, so we have a question from Ernesto, and it's a question that everyone makes. Why do we use Python? Does Python has an advantage over other languages?
Yeah, it's a good question. I mean, everybody's going to have their own answer to this. I'll give you mine. I like Python a lot. I like its syntax. I find it very expressive. I think that what I mean by expressive is that I can accomplish tasks with a minimal amount of code that I find very readable. So I like the way it reads as I write it, and I frequently find myself going, that was just a beautiful way to express do this in code. So I like it for that. It has an incredible built-in, the what we call the standard library, right? The modules that come with Python when you install it is just amazing compared to some other languages. It has an incredible community, a lot of third-party libraries that exist. So, if there's a challenge you have, chances are someone has already had that challenge and probably done it for you, or at least given you some patterns you can follow. I'm not gonna lie, there's some downsides, right? Like it's not the most memory efficient language out there. But I do feel like for every hurdle I run into, there is a way to get around it. For instance, with the memory thing, you could use a C extension. So Python is pretty good at wrapping lower level languages like C. And so for me, it's just like it's the perfect balance between developer time. I can rapidly prototype things. I can get stuff done really, really quickly. And when I do run into sort of maybe lower level limitations on things like multiprocessing or something like that, or memory management, generally, if it's a big enough problem for me, someone solved it with a wrapping a lower level library like C or something like that. But at the end, it's personal preference, and I just really like it.
Awesome, thanks, Sally. So Amir is asking, what's the advantage of Napari above the Fiji and macro programming? So
yeah, I mean, that's such a good question. It gets asked in pretty much every meeting where Napari comes up, and I would say advantage isn't really the word I would use. I would just say, it's in Python. That's probably so. The main things that I think the main current leg up that I wouldn't even call it leg up. The main thing that differentiates Napari at this moment is it's written in Python. It integrates with the Python ecosystem a little easier, and it sort of was designed from the beginning for big data, right? So this the ability to kind of work with out-of-core data is perhaps a little more natural from the beginning in Napari. That's not to say that Fiji doesn't have amazing tools for working with out-of-core data, and Fiji's been around for 20, 30 years at this point, so it's got way more wealth of ecosystem around it. The advantage the disadvantage of Fiji, of course, is that while you can use Python with it, it's hard to use the full Python ecosystem. Like, it's difficult even to use NumPy, for instance, in Fiji, because it uses Jython, and there's some limitations with using non-core libraries in Fiji. Now, one can use PyImageJ, which is, so basically you can use ImageJ stuff in Python, but it's hard to use Python stuff in ImageJ. I don't know if that made sense. Anyway, I think this really comes down to it's written in Python, to begin with, and this multi-dimensional viewer thing is sort of the original design of Napari, whereas with Fiji, it's something that's coming to Fiji currently with some amazing developments. But yeah, that's
awesome. So Chia is asking, like, with the implementation of this technology, was the immediate benefit for biologists compared to previous conventional microscopy work? And a follow-up on that, and I will just read out right away. Which other benefits we could expect for the coming years in this line of microscopy?
Yeah. With regards to the microscopy, the main benefit with light sheet microscopy is, I could summarize it in terms of efficiency of light collection. So to be more concrete about that, well, let me just say, like, it helps. Its main benefit is live cell imaging and fast imaging. So when we're imaging living samples, we are dumping an extraordinary amount of light on them. Like, the irradiance in terms of just joules per second of energy that we are shining onto these cells is much more than like what we, the sun on us on the hottest day. It's like thousands of times more. And cells grow up in the dark, so we're killing these cells as we're imaging them. So anything we can do to be more efficient with, like, shining light only where we need it to illuminate just what we need, essentially can benefit quite a bit. So we can image now perhaps thousands of time points over the life of a cell dividing rather than just maybe 50. So that for me, that's the most exciting thing. It really comes down to 3D imaging of cells over time. If I tried to do that with conventional techniques, I would kill the cell, or I would do it I would run out of, do what's called bleaching the image. So there'd be nothing left to look at after a few frames. And in terms of coming out of this in the near future, I think there are evolutionary and revolutionary changes, and I would say light sheet on the whole is a revolutionary change, but it's not tremendously easy to use at this point. So one of the big things I think we'll see is for it not to have to live in a core facility where you have someone like me to run it. I would really like for these things to be a lot more easy to use. But on the technological side of things, I think that getting deeper is a current challenge. So as we image deeper into cells, light scatters, and so imaging deeper and deeper is another thing that people are trying to do.
Awesome. So I have a question from Ivan. He says, great tool. Is it possible to save complete workflows of the transformations made to the raw data in napari? This is trying to tackle the reproducibility aspects. So this would be great for open science. So yeah, we actually are working on that at some point.
Yeah, I totally agree. Yeah, that's actually, to go back to the Fiji and macro thing. So one of the really nice things about Fiji is it has a macro recorder. You asking the question probably know, but it was mentioned earlier, where someone can do a sequence of steps. It gets recorded as a macro, and then you can just run that to reproduce it. That concept, of course, is awesome. We want that very much. So the answer is right now, no. You would have to have written the script yourself that would reproduce. We would love to simplify that and sort of essentially make something that is the common workflow language for napari. Actually, I will mention a
well, I guess it's worth pointing out that it's coming and it's on the pipeline. It's just a hard problem to tackle.
Yeah, it's a hard problem to tackle. Here's somebody Robert, who's made a plugin that is trying to sort of capture a sequence of actions and in a reproducible way. So challenging, want it, yes.
Awesome. So what library was used to create the graphical interface?
Yep, PyQt. PyQt. We use QtPy from originally Gonzalo, I believe, and others made a a compatibility layer, so we can use either PyQt or PySide. But yes, the GUI is written with Qt.
Awesome. This is a really specific question. Do you know any developed plugins using napari to analyze single molecule localization microscopy data sets?
No, I don't actually, and I think I would know of one. So I think at the moment we don't have any like Storm, Palm, single molecule localization data sets or plugins. One thing I actually have toyed a little bit with converting our points layer into something that would render, like let's say you've done the analysis somewhere else to even just render it in napari. And I mean, you can do it. It's fine. I think there are some things that would make that look even nicer, but that's come up a number of times. It's basically like we're teed up to do that. The performance of the points layer is sufficient. In other words, it just needs a little bit of niceties around single molecule localization, but no, not yet.
I guess it's also a good time just to invite Amir if he would like to contribute somehow to working on this.
Yeah.
Well, I don't know what would be good links to share at this point. Maybe the Zulip link. I'm not sure.
Yeah, Zulip and the core repository link is always a good one. I mean, everything sort of hovers around the napari GitHub repo, but the Zulip link is our chat. Where we're always just kind of there if you want to come ask questions or.
Awesome. So I'm sharing on the comments. I'm sharing the Zulip link, and I'm also sharing the. Where is this? And I'm also sharing the repository. So yeah, you can find all the information you would like to know about the napari project. You can find it on the repository. I think that covers the questions we had so far, Tali. So you may want to continue to.
I will.
Cool.
Yeah, so this next part, let's see, I'm going to try to keep. Yeah, I will keep that over there so I can see it. If you ask questions, I will be able to see them. So this.
Let me jump on you just 1 second. Yes. Could you make that slightly larger?
Yeah, I can.
Like, one more, maybe? Perfect.
Okay. Thank you. Let me. Okay. So I'm not going to walk through this line by line. I will mention actually, Gonzalo, can you put the link to this? This is at the.
Yes, I will.
Okay. Yeah. Basically, this notebook is available if you want to play with it, and there's some sample data in there as well. But this is the example I showed earlier of the yeah. Thank you. Of using Napari with Dask. So just a couple. Let's make sure I'm still running here. Yeah, good. The main object in Napari is the viewer object. So from Napari import viewer equals viewer. This is the main viewer object. Most of the entry points into getting data in there are the viewer add whatever, add image, add points, add layers. So for all the various layer types I was talking about, there is an add underscore layer type method. And so here I'm adding an image. Cells 3D is just some sample data from scikit-image and giving it some parameters. And then I am making some points, so just another layer type, just a random set of 3D coordinates, and I'm adding those. And then programmatically, I can control the viewer state. I don't know, that's really sort of a side point, but by having added basically this image and these points, I get these 3 layers here. And of course, they each have controls that I can use to change the way they look, and I can rotate and etc. I can go back to 2D here and scroll through the dimensions, etc. So that's the real core operation of Napari. These things are just kind of cool to point out that while we don't really have this kind of workflow language, so to speak, to actually perform analysis, it is really important to us to make as much as possible open programmatically, so that you can control all of that stuff that you would do with the graphical user interface programmatically. Sometimes we miss things, but that's certainly the driving principle there. So this little bit here is just about Dask. So I talked earlier about how Dask arrays are like NumPy arrays, but they are lazy in the sense that you can create a massive Dask array instantly, and it doesn't actually do anything until you call that compute function. So I am going to connect to a Dask client here, and I'm going to do that so that, actually, did I lose the... They're over here. Gonzalo, let me know if I've made anything too small again to see.
I think it's working great, and for the rights, it's just for showing, so I don't think we need to make it bigger, okay. Right?
Okay, so what I put on the right here is just a little view into what Dask is doing. You're going to see tasks getting executed here and the progress of them up here. It's only there just to show you what's happening behind the scenes and when things are getting computed. But for instance, I'm going to import from Dask array, or I'm going to import array from Dask as DA. This is the sort of standard way to do it. And from Napari, I'm just grabbing a convenience function called viewImage. ViewImage just makes a viewer and adds an image to it all in one go. And so here I'm going to make a massive, giant, multidimensional Dask array. And Dask has a nice little repr for this, so I can see that I just made a 1 terabyte array. Obviously, it didn't actually render that in real time because that would have taken a lot longer. But this object, massive array, represents basically something that, were I to load it all, would be 1 terabyte. But it's broken up into little 8 megabyte chunks, and that chunk shape is shown here. So it's basically individual 1K by 1K, 1000 by 1000 planes in multi-dimensions. And if I take this massive array and I pass it to Napari, viewImage massive array, you might have seen a little bit of activity flurry there, and then this window pops up. Now I'll move this over here. The point is, it has only computed that first plane, because that's all I needed at the moment to show it. And if I start moving this slider around. You know what? Yeah, it's not task graph I actually wanted. Hang on, I want the I can never remember which one I want. I think it's the task stream. Yeah. Okay, so you can see the jobs flying by here as I load individual planes. So if I stop this and I go, each time I move this, it's doing that calculation in the background. So this is all Dask being amazing. All Napari has to do is basically be careful about indexing to make sure that we don't load the full data sloppily, basically, like on accident, and Dask will kind of take care of the rest. So that key idea there, and actually you'll notice if I click the 3D button here to load the full volume, there will be one big flurry of activity because it needs to load everything from that volume. So right there, it had a big loading thing. And if I go to another time point, it loads that whole thing. So that's Dask basically doing its work in the background as Napari moves around. That particular feature is really awesome. And most of what I'm showing you here, that is the core concept right there. And so everything I'm showing you follows just that if we make a Dask array, and if it's properly chunked in the sense that when we chunk the array, we're telling Dask how to break it up into little bits. And if we do that incorrectly, we can accidentally load too much at any given time. So if we've done our job in chunking it correctly, then Napari can do its job in loading just what's necessary. And then the next two key concepts from the Dask side that we can take advantage of in Napari is Dask delayed and Dask map blocks. These are two concepts or functions from Dask. Dask delayed lets us basically convert any function to something that is lazy in a sense. So if you have a function, let me just jump into the example. So TIFF file imread is a function from imread is a function from the TIFF file library that takes a path to a TIFF file on your hard drive and reads it and returns a NumPy array. It does it immediately. It immediately loads it into RAM and returns it to you. I can take that function object and wrap it in Dask delayed. So this is like a decorator that I'm not using as a decorator. I'm just immediately calling it on the function. What I get back is a version of the function that when I call it, it doesn't actually run it immediately. It returns to me a delayed object. And if anybody's worked with JavaScript or anything, this is the concept of a promise, or if you've worked with multiprocessing in Python, it's like a concurrent future object. It's an object that represents some future task that may eventually happen. So that's what lazy mread now is. It's like a function that can be delayed. So I'm not going to go into this function, but basically create Dask stack here is, I'll scroll down to the usage. It's a function that takes in a path right there. That's just a local, actually I'll show you that on my hard drive so you can see it. It's this. This is kind of how a raw data set might look coming off of the microscope, just, thousands of files. Each one is an individual 3D stack. So I can just give it a pointer to that folder, and it will look through that folder here with the glob and just look for all the TIFF files in there. And then for every file in that folder, it's going to call this lazy mread function, that delayed version I talked about, on that path and return that delayed object, that kind of concurrent future object that represents some eventual data. It's kind of meta, and it's kind of hard to wrap your head around at first. And then we can convert that delayed object to a Dask array. And then we'll just stack all those up and return it. So if I run this, you'll see it returned almost immediately. So I haven't actually read anything, but what I have now, this object stack, is a Dask array, and it represents a 2 by 600 by 65 by 256 by 256 array. So this is a 5-dimensional array where it's basically assembled all of those files in the in the dimensions that they should be based on my function, but hasn't read anything yet. So now I can take that stack, that DAS stack, and oh yeah, somebody is noticing Dask array comprehensions. Yes, Dask array can do. Well, basically, I'm doing a comprehension here and then stacking it with Dask stack, but it can handle that too. Right, so let's say I'll then just take that stack, pass it to Napari with the view image, and then I'm just making it into a 3D viewer. This screenshot bit here is just so that you see a screenshot in the Jupyter notebook, but let's see what happens here. Oh no, what's happened? Ah, there we go. Okay, so it loaded it. And again, if we watch the task stream over here, we should see stuff happen, right? So actually, I'll play it here because you might actually be able to see, they're flickering, but you can see there's a get item and there's a imread. So at each time point here, it's using that TIFF file imread function to load that particular 3D volume, and then it's returning just that plane to me. Now, if I stop here, you'll note that this is kind of a mess, right? Like it does not look like biology. It's all kinds of weird. And this gets to the sort of core domain specific thing here, which is that I keep alluding to earlier, which is that there's some pre-processing required. This is kind of interesting if you're curious. So I'll explain why this is. And if you're not curious, I guess I'm explaining it anyway, aren't I? So
yeah, go for it. This is pretty cool anyway.
Okay, this is a zoom in on basically the acquisition in the microscope. So I showed you that big picture before. The way this, if we kind of zoomed into where the image is actually made, there's two objective lenses, and they're at 90 degrees to each other. One of those lenses is forming the sheet. So this is the excitation objective, which is forming a sheet in space. And the other lens is gonna collect an image of whatever gets excited in that little area. Now the sample sits in a cover slip here, and it sweeps through the sheet. So basically, this is how an image would be collected. We have the stage can move. It can move in all axes, but basically the way the fastest motion is this way. So it's like, here is our image field of view. And as we move the sample to take a 3D stack, you can see that effectively each image is a little bit offset from each other in the third axis, in the z-axis. And so on a conventional microscope, that's not the case, right? Your z-axis, as you go through focus, would be orthogonal to the image plane. Here, it's at a 32-degree angle to the stage motion. So that is a little unusual. And if we sort of, if we just opened that image stack in a conventional image viewing program like Fiji, if you don't tell it any more information, or Napari for that case, it doesn't know that these are off like that. So it's going to assume that they're on top of each other, like what I showed there, right? So in real space, the images were taken like that, but when you just open it in a image viewing program, it's going to skew the image. And so we basically need to de-skew the image to shift each plane in the stack back to its position in real space. So this is just a basic affine transform. It's computationally pretty straightforward, but we now have to do this to every volume that is collected. And then optionally we can deconvolve again, which would just be yet another step in the processing pipeline. So the point is that all has to happen before we can look at it. Otherwise, it looks like what you just saw a second ago, which is hard to interpret and even harder to analyze. So right, so that's the challenge, is that not only do we have a lot of data that we would like to load lazily, we also want to do some processing. And this is, I alluded to this earlier, my original strategy was just to do all that processing up front, to say, okay, I've got a data set, let's just de-skew everything, do whatever I need, and now I have a duplicate that is ready to analyze. But like I said, now I've gone from something like 50 gigabytes to something like 500 gigabytes. And if I'm going to save that for every data set, and if I'm collecting 5 of those a day, you know, it's not like impossible. It's just really, really wasteful, right? And we can do better. So this part is about queuing up that computation a little lazy, so that even not only does the loading of the data happen as I move the slider, but so too does this deskewing operation. And really, this could be a proxy for whatever it is you need to do to your data between loading and viewing. You can tell Dask not only, you know, load it, but process it lazily. So the way that works is with map blocks, Dask array map blocks. This is a pretty cool function. Map blocks essentially just maps some function across all of the chunks in your Dask array. So here I'm using this is not important for our sake of our discussion, but this is just, I'm getting a, I'm seeing a question, are there side viewer easily available in the napari viewer? I'm not sure what side viewer means. Do you mean like an additional viewer? I'm going to keep going, and you can maybe clarify what you mean by side viewer.
Yeah, maybe it's more canvases, more viewers. I don't know.
Yeah. I mean, if you mean, can you make multiple viewers? Yes. If that's not what you mean, please clarify, and I'll get back to you. Okay, so I'm just getting a deskew function here. You can ignore this chunk. The point is, like, deskew is now a function that will take a 3D array and do that deskewing that I was talking about to see it in real space.
You take a look at this question.
Yeah. So if I understand this, it would be possible to have a fully automated deskewer for specific microscopy technique. That would be so nice. Yes, that is basically exactly what that's what I'm sort of building up to, and that you could wrap all of what I'm showing you here in even a plugin, which I have for Lattice, but you could generalize that too and make it so that basically you can encapsulate all of the deskewing and everything you need to in some plugin perhaps, and side view, sorry, side view, got it, and have it on demand. So Yvonne, yes, that is the goal. Amir, side view, orthogonal views, that is a super high demand thing. We don't have a natural like orthogonal view, ortho viewer in there yet. Such an obvious thing that we need. There are some underlying data structure challenges that we want to solve before we tackle that, but suffice to say, it's like high up on the list of things we want to do. So to get back to this map blocks thing, deskew is my function, takes a volume, deskews it. What I do here is I take stack. Remember, stack is my Dask array. It's my Dask array object encapsulating the whole experiment, and I call on its map blocks method deskew. And I have to tell it something about the data type and the chunks, the chunk size of the new of what will happen after I deskew it. Because actually, you probably noticed that if I scroll up here, deskewing changes the shape of the data set. You know, it actually makes it larger. So it's gonna change things. So when I run this stack map blocks deskew, if I run that. You'll see it's actually telling me that the shape is going to change. The X dimension gets larger. And it returned immediately. So it didn't actually do any deskewing. It just returned to me a new Dask array that has encapsulated the concept of skewing this data when I ask for it. So I can now take that new Dask array and pass that to Napari. And let me run this. Here we go. And now, now it looks right. Now if I rotate it, it looks exactly like it should. It's not all skewed. And if I run play here, you'll notice now, in addition to imread, we have this noisy deskew. I called it noisy deskew, I think, because I put some print statements in there or something. I don't remember why I did that. Anyway, so now not only is it reading the data, but it's deskewing it. So that I can rotate it as I'm sorry, so that it's in correct real space coordinates. Now, again, just to point out, like, obviously this is playing a little slower than it probably would if I had all this done up front and greedily, but I can save all that space and not have to do that. So that is a pattern that I use a lot in Napari. I think is pretty nifty. And actually, let me try to show something here. I didn't. This gets to Yvonne's question. So let me hide this notebook now. And
we have a question there. Is this skewing like people do in oblique plane microscopy optically?
Yeah, absolutely. Yeah. So if you're talking about this new thing from Redo, where there's a new approach where you can sort of do deskewing optically rather than mathematically. So yes, it is. The fact that they can do that optically is pretty cool. Each has a slightly different application because I believe there's some projection that happens there too. But anyway, yes, to answer your question, it's referring to the same concept that basically in both cases, we are collecting the data in a somewhat unconventional way that leads to a geometry that is abnormal and a little unusable. And it's skewed specifically with respect to real space. So it's the same challenge that they have in oblique plane microscopy where they have to deskew it. Let me, as a final thing, see if I can get this to work. So I'm going to open Napari. I'm going to see what plugins I have installed because, okay, so NDTIFFs is a plugin that I wrote to basically do everything I just showed you in this notebook, but as a plugin that will recognize when I drop a folder, that's a lattice light sheet folder, let me just treat it as such. So the way it will know that it is a lattice light sheet folder is because somewhere in this folder right there is a text file that is a very specific metadata. And so basically my plugin says, okay, if you give me a folder and it has a text file _settings.txt and maybe has a certain type of metadata, I recognize that as a lattice light sheet data set. And I will open it as such. And you can install, just a side note here, you can install multiple readers in Napari. And when a user drags a folder or an image, we basically go down the line of readers, giving each a chance to say, yes, I can read that. And then, of course, the user can order those as well to override any of that. But let's try this. I'm going to just actually, if I take one image in here, you can see this looks like it's moving from left to right. And if I go into 3D, it's all messed up. So this one image has not been deskewed because it does not have the information necessary to deskew it. That's all in that settings text. But if I give it the whole folder now, which is 10 gigs, I believe, it opens nearly immediately, and it has done all that deskewing, and I can scroll around to different time points. So the point there is just that you can capture all of this kind of Dask stuff in a Napari plugin. I mean, first of all, I would say Dask is amazing. You should probably look in. You can use it all over the place. I encourage anybody working with like large data to look into Dask, but you can also use it in a Napari plugin. That was what I had to show you. So I will stop there and take any more questions that may come up.
No, just some praise. So I think it's worth saying it. So just mind-blowing, amazing stuff, Tyler. Thank you very much.
Thank you, Julian. Glad you liked it.
Actually, Julian gave us a talk about using Dask for machine learning applications. So he's very excited to see Dask used in other contexts.
Cool. I wish I had seen that.
So is there anything else you would like to point us? Otherwise, we can just start to go. I mean, we changed the format a little bit today. We started doing the questions as the things were going because I think Tali feels more comfortable doing that. So yeah. we just changed things a little. So Julian is asking, are you running this on only 4 cores, or what are you using as? What's your machine? I guess.
Yeah, I have. Yeah, it's a 4-core. It's 4-core CPU. I can't remember which. You know, I have Hackintosh here. I can't remember what my CPU is, but it's not terribly new. Yeah. I mean, so most of the, you know, like, I think actually that's a really, this touches on some interesting things. I think that, like, for a while, I was thinking, okay, I need bigger and more monstrous, like, computers, you know, and we did. When we got this instrument, we bought, like, you know, we bought a computer that had 512 gigs of RAM and 48 cores, etc. And you see a lot of people doing that. They crank it up because, you know, and obviously that's useful. If you have it, you should use it. But I think I'm also excited to just kind of make things lazy and smart, so that I don't have to rely on that massive beefy thing at work. Is it possible to use this algorithm to analyze blood? I don't know. I am not sure how to answer that. I don't really have an algorithm there. The deskewing wouldn't. Yeah, I don't know how to answer that.
Yeah,
if you have an algorithm that analyzes blood and shows images, you could probably wrap it for napari.
Okay, cool. Well,
proteins, sure.
so yeah, I guess I have some questions, and it's so everyone working within this project, I'm really happy to be able to collaborate here. Everyone has their pet peeves on what they want to work on and what they want to do first. Disregarding maybe whatever the needs are of the community, which isn't a bad thing. So I'm curious, like, what's your pet peeve in this project? And it's like, this is something I want to get going, and I will push it myself if I have to or something. I don't know. I'm just curious.
Yeah, I mean, well, so I think ortho viewers is about as good of an example of something that we, you know, as an application that purports to be good at 3D, you know, viewing, to not have an orthogonal viewer at this point. Like this ortho viewer would be the thing, you know, that simultaneously shows you X, Y, Y, Z, and X, Z. And for medical imaging, that's, you know, it's like, you got to have that to even kind of, you know, so that's a huge one. I haven't personally, you know, this is not the answer to your question because I haven't, like, taken that on myself.
Is there a technical issue here because the things are being calculated lazily to generate these planes? You need to perform more calculations, or what's the main
so the big hurdle
challenge?
the big hurdle for orthogonal viewers is the way that we, without getting too in the weeds, it's the way that we slice the underlying volume. So if you have a 3D volume, we basically have slicing on the layer. So the we need to make it more natural to have multiple views onto the same data set than we currently do. So we basically have, like, this is the view onto the data set. You can view it in 3D and you can view it in 2D, but you can view it in 3D or you can view it in 2D, but you can't view multiple slices on different canvases at the same time, because currently that slicing operation, the operation that goes into the 3D data and extracts a 2D plane to show you, is, as far as we're concerned, in the wrong place. We need to, like, basically move that operation so that slicing comes off of the data, the layer data.
Awesome. There is a question here, like, 3D visualization is based in a C++ library, or is it a Python library?
Yeah, so the 3D visualization, again, that's VisPy really doing is the workhorse there, and that wraps OpenGL. So you could basically call it, I mean, it is compiling things down to GL at runtime. So it's not a static C++ library, but it basically helps you write and compose, like, so shaders are the names of, you know, shaders and fragment code are the names of little programlets in OpenGL that execute the 3D visualization and render things to screen. And so, yeah, I hope that answers the question. The point is that basically VisPy lets you sort of dynamically compose OpenGL code, compile it, and then run it at runtime. It's sort of like a just-in-time compiler.
Awesome. Okay, This is great. We're almost near the end of our 1.5 hour plan, so I think the timing was great and the amount of questions was perfect. So
yeah, thank you all for your questions. I appreciate it.
Thanks a lot, Tally. Oh, we have more questions. Okay,
Glue
Ivan, keep them coming. You always ask questions. Come on, keep them coming. Are you familiar with GlueViz? Any plans of adding that kind of interactivity? So I think it would be important here, Tally, sorry I jump on, is we have talked a lot about microscopy today, but to be frank, Napari is completely agnostic in terms of what it wants to achieve. It just happened to focus on this group
yes.
at the moment. So if you could expand on that maybe while answering, that would be great.
Yeah, 100%. That is actually, like, a lot of us come from the imaging sort of side of things, but it is important to all of us that it not be like too narrow in scope. So geospatial, stuff is a natural fit. Astrophysics is a natural fit. We'd love to sort of get those communities more involved, mostly so that we don't, make decisions that narrow the scope of this unnecessarily. I have heard of GlueViz. I'm afraid I don't know enough about it. So this is like a perfect example of me wishing,
So I guess, so I guess if you scroll down, you probably see some examples.
Am I sharing my screen still? Oh yeah, I am. Okay.
Yeah, you are.
Is it geo stuff for the most part, or?
No, actually, I think that glue is very heavily used in the astronomy world, but this kind of interactivity, I guess you can pause there, and it's like you have multiple views of things, and you can represent the data in form of plots to the right. So if you could trace a plane, you could make a line or a profile plot, and maybe get some statistics. So I think we have some plugins already that are trying to do that, the profile plot and all that in terms of interactivity.
Yeah.
So maybe not to the level to GlueViz is currently doing, but I think that we are on our way to do something like that.
It is conceivable that I won't even say. Yeah, I'm not sure. I was gonna say it's possible that some of the VizPy developers have connections there, but I don't know.
So yeah, I mean, we need to connect with other communities, and of course, GlueViz has their own niche and their own use cases, and their own needs, of course, and why they were created. But I don't know. I think that's something to work on in the future. We're still not actively talking to the GlueViz community.
I agree.
So there is another question by Amir, and it's this is about Dask specifically. So any command like np as type to convert the type of a variable in Dask, does it act the same in terms of, I guess, delaying the computation, or?
Yeah. So in this case, the answer, I believe, is yes, because the np just added dot to the values on an array. Yeah, I mean, if you're asking, basically, can you treat these Dask arrays like with many of the things you could do with NumPy, like cast them to a different type? Make sure I understand to convert the type variable. Yeah, the answer is yes. Not only that, but you could do all the standard like max, min. Actually, if we go to the Dask array.
So most of the API it's available in Dask, but there are some things that are not there, I guess.
Right. That's right. So this page here, am I showing? Yeah, I'm showing. Okay. This page here shows you like the subset of the of the API that is covered, and as type, I believe, is definitely in there. But there's quite a bit that's covered already. It's pretty awesome.
Cool. So last 2 minutes. Okay, so Julian says that will be included on the graph of computations for later execution. So he's our resident Dask expert. Cool. Thanks, Julian. And thanks a lot, Amir, for all the questions you made. So I think we're close to an end. So I think it's fair enough to start stretching and preparing for lunch or whatever your plans are after this meeting. I would like to thank again Mafe for all the hard work in helping organize these meetups, and of course our guest Tally for that awesome presentation. I always enjoy your presentations and you explaining stuff. So
thank you
thanks
so much for having me. It was a lot of fun. I really appreciate the questions. It was great being here.
And just so that you know, we'll keep this format and we'll try to go forward with this as far as we can take it and as far as we know people that are willing to do it. So I would like to just remind you that follow us on the different social media outlets. We had a thing with Instagram that we kind of lost access to our account, so we created a second account in the meantime while we can gain access. So just let me fix that. I'll just put it there. So yeah, the Instagram account, the backup account is now like Twitter, so just PyBook. And in the meantime, we'll try to fix that. Otherwise, we'll have to migrate to that one. We'll keep doing this. There was something I wanted to share with the community. I'll probably share it now with the people in here, but also we'll be sharing that in the different social media platforms. And I'll start having a couple of, maybe an hour or 2 half-hour slots during the weeks to try to do some coaching and mentoring in whatever topic I am capable of coaching. So if you want to learn more about Python or TypeScript or software engineering in general, or Napari and open source, or working with communities, whatever is your interest, even practicing English, if that's something you would like to improve, or just maybe improving your LinkedIn or whatever. I don't know. It's going to be pretty open. It's an experiment. It's part of giving back to the community. I mean, all these projects have been very good to me. So I'm trying to give back to the community. So hopefully they can join and collaborate and participate. And hopefully you can do the same at some point when you have some time. So I wanted to share that. I don't know, Mafia, are we forgetting something? We have our next event by the end of September, I think, the 20-something.
Yeah.
It's going to be Maria Camila, I believe.
Yep.
Okay.
September 23. Yeah.
Cool. So that's our next event. It's a Thursday evening, like the other time frame we use for these events. And we'll share that the example notebook was shared on the comments, but we'll also share it on the description as we usually do. And yeah, so yeah, Julian asking. So yeah, there is, I think, a repo. So I'll share that on the description, and we'll share it also in the different channels we have. So once again, thanks, Mafe. Thanks, Tally. This was awesome. And hopefully we can see you in a couple of weeks.
Thank you.
Okay.
Thank you so much, guys.
Thank you.